Alright, section 6.2 talks about the different growing environments and different growing conditions for microorganisms. Now, in this first objective for the chapter, you have a variety of vocabulary, which a lot of these will pertain to this section. And then figure 6.5, looking at the temperature requirements and therefore what organisms might be called or what group they're found in based on the temperature requirements. The terminology of organisms living together in a positive or negative manner. How well do they work in partnership with each other? And then also the idea of biofilms and quorum sensing. So there are a lot of different environmental factors that influence microbes, specifically in terms of their growth. And in the environment, organisms are exposed to a wide variety of factors that are, in addition to the actual nutrients. So we've talked about the nutrients, the chemicals, and the main issue is that these environmental factors will alter the function of metabolic enzymes. All right, enzymes are the protein doers of the cells, but they also require specific environment, and that is mainly going to be temperature and pH. And if the temperature changes or the conditions change in the environment, it's going to be dependent on how well the organism can adapt through their enzyme system to be able to survive. So with temperatures, organisms can be classified based on where their maximum growth is going to occur. All right, so on the y-axis in this figure, you have the rate of growth, with more growth being as you increase on the y-axis, move away from that point of 0. And then on the x-axis, you have temperature in degrees Celsius. So there are 5 major groups presented here. In the blue, you have the psychrophils. Those guys like the cold temperature, and their maximum growth rate is going to be right at about 5, 6, 7 degrees Celsius. And their minimum temperature that they can actually replicate at, although it's very slow, is going to be -20. And then the maximum is going to be 15. And that's what you see with the red dots and the red and the blue dots on all of the different growth curves. The yellowish orange are going to be the psychrotrophs. And then the green are the mesophils. And the optimum growth there is going to be right at about 37 degrees Celsius. So that's going to be perfect for growth where? Growth in humans. Then the thermophils love heat, and those are going to have a most efficient growth right at about 70, 72 degrees. And then there are the extremophils. All right, the extreme thermophils, they are extreme in their love for heat, and so those are going to grow at like 125 degrees Celsius. That's major. All right, so what the terms that you want to be aware of for this figure are going to be the psychrophils, knowing that those organisms grow at cold temperatures, the mesophils, knowing that those have a maximum growth rate at 37 degrees temperature, 37 degrees Celsius, and then the thermophils, all right? Not necessarily distinguishing between thermophils and extreme thermophils, but just that thermophils love heat. Now, the one important feature to remember is these mesophils, all right? The fact that they grow at optimum temperature at 37 degrees means what? That means they're going to cause human disease. So these are responsible for causing human disease. Now the other guys, the psychrophils, the psychotrophs, the thermophils, they don't necessarily cause disease in humans, but they can still cause problems for humans because they can alter the environment or grow in different areas within the environment and spoil food, spoil crops, spoil a variety of things, spoil consumer products. So they all have their place, good or bad, within human life. All right, now, as far as growth requirements, you can also look at oxygen. Yes, oxygen is one of those major elements that organisms are going to use to grow, but their growth pattern can also be used to categorize them. So up top here, you have the aerobes. Right? Aerobes are going to utilize oxygen. And in these test tubes, at the top part where you have the coat or the cap that is closing the tube, all right, you have higher amounts of oxygen here. And if the tube is stationary, then as you go down this tube of medium, all right, you decrease the amount of oxygen so that down here at the bottom, assuming you aren't shaking this tube, there isn't going to be any oxygen. So if an organism is an aerobe, it's going to be right up top because that's where the oxygen is. An obligate aerobe means that it is obliged to live in the presence of oxygen, which means you're just going to get a very thin layer of growth at the top, because right at the top is where you have oxygen crossing over into the medium. All right, down here you have a microaerophyll. So here they don't grow at normal atmospheric concentrations of oxygen, but requires a small amount of it. And so you have no growth right here at the very top. Maybe hard to see that. But there is some medium on top of this yellow section. And so down here you have a little bit of oxygen, a little bit, and then right down here is where you start decreasing oxygen altogether. So microaerophils, they like a little bit, small amounts of oxygen. Facultative anaerobes, if it is an anaerobe, we'll go down here. All right, anaerobes grow without oxygen. All right, so now you have growth down here where there isn't any oxygen, and it might be the figment of my imagination, I'm not sure, but to me it looks like they've tried to color the tube so that it's more yellow down here, and the yellow decreases as you come up the tube because, yes, there will start to be some amount of oxygen excuse me, being present in that medium. The facultative anaerobes means that the organism doesn't require oxygen, right? It's an anaerobe, so it can live without. But the facultative means that it can still grow in the presence of oxygen. And that's why you see growth all the way through this medium. The aerotolerant anaerobes mean they can tolerate oxygen. And so they don't use it, but they grow in the presence of it. Very similar to the facultative anaerobes. The facultative anaerobes will actually use it, whereas the aerotolerant, they're tolerant, but they don't use it. So that's why the facultative anaerobes and the aerotolerant anaerobes, the picture looks different, even though the bacteria actually are different. So some other conditions that are loved because of the "phil" in the name, or preferred. Capnophils are organisms that grow best at higher CO2 concentrations. A human example of this is going to be Streptococcus. Streptococcus pyogenes likes CO2. All right, and if you want to try to grow this organism in the laboratory, you have to add CO2 to its environment. Add an additional 5% generally of CO2. All right, then the acidophils. These guys love acid. So if, well, never mind. The alkalinophils love base. So they live in alkaline or basic conditions. So the pH is going to be greater than 7. Halophils prefer high salt. And there's a range of halophils. Those that can live in the presence of maybe 5%, 4% salt concentration, upwards of 20% salt concentration. That's really huge. That is a high amount of salt for any living organism to live in. And then human example of the halophils is going to be staph. Staph likes salt. Where do you see staph a lot at? Well, one organism or one species is Staphylococcus epidermidis. It lives on your skin. And when we sweat, that is one way that our bodies get rid of excess salt, or just salt in general. And so the staph organism being on our skin is used to that high salt concentration, and so they like it. Barophils, they like high pressure, and it's interesting, these guys actually need pressure to actually keep the integrity of their cell wall together. It's hard for scientists to study these guys because you find them in areas of high pressure, like at the very bottom of the ocean. And if they try to pull those organisms up to the surface of the ocean, well, as you pull it closer to the surface, you lose the pressure of the water. And their cell walls just disintegrate, and the organism is dead. So the scientists have figured out ways to add pressure to mimic the environment, and so they can study those. All right, then association between organisms. How these organisms live together. There are symbiotic relationships. So organisms live in close nutritional relationships, and they require one or both members. Underneath symbiotic, there are 3 different groups. You can have mutualism, commensalism, parasitism. Mutualism is where both members benefit, so they're dependent on each other. Commensalism, the commensal benefits, the other is not harm. So a lot of the organisms in our GI tract are commensal, all right, because they benefit, or the ones on our skin benefit. They get a home, they get food, but they don't do a whole lot for us. For sure, the ones in our GI tract, there's a lot of mutualism going on there. Both members benefit. We benefit from them digesting our food, and some of them give us different nutrients, vitamins. And so that's a mutualistic relationship. And then parasitism, the parasite is dependent and benefits, the host is harmed. So if an organism from our GI tract gets into our urinary tract, right, that causes an infection, and that organism that was once, excuse me, that was once living in a mutualistic relationship is now a parasite and causing us harm. And then there are the non-symbiotic organisms. These guys are free living, and you can have ones that are synergistic, and so they cooperate and share nutrients. There are ones that are antagonistic, and some members are inhibited or destroyed by others, and this is generally going to be through the production of different chemicals or antibiotics. Then there's gases. All right, and we've already talked a little bit of gases. The important ones are going to be CO2 and oxygen. Oxygen is the most important one when we talk about microbial growth. And part of it is because oxygen can be, in the different processes of metabolism, be broken down, and you can have different radicals that are produced and different negative oxygen species. And so organisms are sometimes able to detoxify it, and then they can live in it. Some are not. And so that's important to remember. All right, and I think this is where we're going to stop, and we'll pick up in a second part of 6.2, starting with the biofilm section.